Hello students, welcome back to your botany class once again. So in the previous classes we have discussed about how protein synthesis starts with transcription, and we also have discussed a little bit about genetic code and the salient features of genetic code. And so today I would like to discuss with you about the types of RNA, because these different types of RNA, they are going to be important in the subsequent process of protein synthesis. We've discussed a little bit about like protein synthesis consists of transcription, then translation. So during the process of translation, the types of RNA, particularly tRNA and ribosomal RNA, which are some types of RNA are to be involved. So today let's discuss a little bit about these different types of RNA before we go into translation. Okay, in the last tutor, in the beginning of this chapter, of your chapter 6, molecular basis of inheritance, we have discussed about DNA, how a DNA looks like, the molecular structure, and accordingly, side by side, we can even discuss with RNA. So RNA is a single-stranded polynucleotide, ribonucleic acid. It's a single-stranded polynucleotide ribonucleic acid, and it's found as a genetic material in most of the plant viruses and in some of the animal viruses. So unlike DNA, know that DNA is a double helical structure, but RNA is a single-stranded. However, this single-stranded RNA undergoes intramolecular folding because of intramolecular base pairing. So they exhibit twisting and folding, giving rise to secondary and tertiary structures. Okay, and like DNA, it's also composed of nucleotides. It consists of phosphate, it consists of a sugar, and the sugar in RNA is ribose, while in case of DNA it's deoxyribose, and it also consists of the 4 types of the bases. In DNA, the bases are adenine, guanine, thymine, and cytosine, while in case of RNA, the bases are adenine, guanine, and thymine is replaced by uracil in RNA and cytosine. So adenine, guanine, uracil, and cytosine are the 4 types of bases found in RNA. And the bases, the phosphate, and the sugar together constitutes the nucleotide, and they form the monomeric units of RNA. Okay, so this RNA acts as the genetic material in organisms where DNA is absent, and it's of different types. And these different forms of RNA, they perform different functions at different level of protein synthesis. So in the last class, when we have talked about transcription, we have talked about mRNA, and we've talked about mRNA. And besides this mRNA, the other types of RNA are transfer RNA, tRNA, it also called as transfer RNA or tRNA, and ribosomal RNA. If you have your textbook, you can open up your textbook and follow me with that. Okay, so in the last class, we have discussed about messenger RNA or mRNA, how it is transcribed from DNA template, how this messenger RNA is transcribed from DNA template, and we have seen how this RNA transcript undergoes processing to get matured, active, and stable. So methyl guanosine triphosphate is added at the 5' end and a polyaethyl at the 3' end to make it stable, and it also undergoes splicing, removal of the introns, which are the non-coding regions, and there are intervening sequences in between the coding regions. So this mRNA or RNA transcript undergoes capping, splicing, and polyadenylation to make it more stable, to make it active and matured and ready for translation to take place. And this messenger RNA, which has been transcribed from DNA, it consists of all the information that was present in DNA. So DNA is the master copy, while this mRNA is the blueprint of the genetic material. It contains all the information of the genetic material, and which has to be translated into proteins, which ultimately determines the phenotypic character of an individual. So this is one type of RNA, messenger RNA, which is transcribed from DNA template or from the genetic material. Okay, The next type of RNA is the transfer RNA. The next type of RNA is the transfer RNA, or it's also known as tRNA. Okay, so all these types of RNAs, be it messenger RNA, tRNA, or ribosomal RNA, they are synthesized from the genetic material. They are synthesized from the DNA, and they undergo processing, they undergo folding to give rise to this tRNA, or to the ribosomal RNA, or even this messenger RNA. Okay, so this tRNA is involved in protein synthesis during the process of translation. And as you can see, or as you open up your textbook and you look into the figure of transfer RNA, you can see that it has got 4 arms. Okay, but before that, this tRNA, it is a polynucleotide chain of about 75 to 93, a polynucleotide chain of 75 to 93 nucleotides. It consists of about 75 to 93 nucleotide bases, and it undergoes intramolecular folding, twisting, giving rise to this T-shape, giving rise to this clover leaf structure. Okay, so this tRNA constitutes about 10 to 15% of the total RNA of the cell. 10 to 15% of the total RNA content of the cell, and it consists about 75 to 93 nucleotide bases, and its sedimentation coefficient is 4s. And because of the intramolecular base pairing or complementary base pairing, this tRNA polynucleotide chain, it undergoes or exhibit secondary and tertiary foldings, and thus taking the shape of clover leaf. Okay, taking the shape, the secondary folding of the tRNA resembles a clover leaf, and this clover leaf model was proposed by R.W. Holley, and because of which he was awarded the Nobel Prize with Corona and Nuremberg. So the folding of the twisting and folding of the RNA molecule give rise to the resemblance of a clover leaf, and it was first proposed by R.W. Holley. And also, this tRNA molecule, they undergoes tertiary folding. They undergo tertiary folding, and it gives rise to a 3D structure which resembles an inverted L shape. So the 3D structure of tRNA resembles an inverted L shape, and the 2-dimensional structure of tRNA, it resembles a clover leaf. And so these are the 2 models of tRNA, clover leaf model and the 3D structure of tRNA. Okay. So let's look into the clover leaf model of tRNA. Okay, so this clover leaf model consists of 4 arms. They consist of 4 arms, and 3 of which are loop-like, and 1 is a stem. 4 arms, 1, 2, 3, 4, and 3 of which are loop-like, and 1 is stem-like. And the stem arm of tRNA has got both 3' and 5' ends, and the 3' end of the tRNA has a hydroxyl terminus, while the 5' end of the stem has a guanine monophosphate terminus. So the 3' end of the stem, or is also known as the accepted arm, ends in a base sequence of CCA, and the 5' end of the acceptor arm ends in guanine. So why is it called as an acceptor arm is because that this arm receives amino acid. This arm is responsible for receiving activated amino acids during the process of protein synthesis. During the process of translation, this information, the codons which this mRNA which codes for specific amino acids are to be read by this transfer RNA, and accordingly the activated amino acids are brought from the cytoplasm, and they are joined together, connected together by peptide bonds. So this is the arm, the 3' arm, or this acceptor arm is responsible for receiving the amino acids from the cytoplasm and bringing them and connecting them during the process of translation. So this acceptor arm is a stem of 7 base pairs, and ends in a base sequence of CCA, and this cytosine adenine arm or the A arm, this base sequence A receives the amino acid. Okay, the next arm is the DHU arm. The DHU arm is also known as dihydroxyuridine arm. So this DHU arm is an arm of 4 base pairs, and it also ends in a loop, and this loop region is an enzyme recognition site, and this is the site where it binds to amino acid activating enzymes during the process of translation. Then the other arm is the anticodon arm. The anticodon arm is an arm of 5 base pairs, and it ends in a loop, and the loop region of the anticodon arm has a definite sequence of 3 nitrogenous bases, and this sequence of 3 nitrogenous bases is known as the anticodon. And so the purpose of this anticodon is that it is able to read the mRNA. It is able to read the codon sequence on mRNA. This anticodon, which consists of 3 nitrogenous bases, reads the codon on the mRNA, and accordingly they bind to the mRNA temporarily and brings, and as such this tRNA brings the correct amino acid for the formation of peptide bond. So this anticodon reads the codon sequence on the mRNA, and this is known as the anticodon arm, which is a 5 base pairs arm. Then the next arm is the ribothymidine pseudouridine arm, cysteine arm, and this ribothymidine pseudouridine cysteine arm is an arm consisting of 5 base pairs. And so like the DHU arm and the anticodon arm, it also ends in a loop. And so this ribothymidine pseudouridine cytosine arm is associated with the ribosomes. And these ribosomes, be it in prokaryotes or in eukaryotes, these ribosomes are the sites of protein synthesis. So this C arm consists of this sequence of ribothymidine, pseudouridine, and cysteine arm. They are responsible for binding to the ribosomes during the process of translation. Okay, so these are the 4 arms of tRNA, and this is responsible for reading the codons on the mRNA by temporarily binding it to it and bringing the activated amino acid from the cytoplasm. So its important function is during the process of translation, which we will be discussing in the next few classes again. So this tRNA resembles a clover leaf in its secondary structure, and it resembles an L-shaped structure in its tertiary structure. So this is also a type of RNA, and it constitutes about 10 to 15% of total RNA content of the cell. Okay, so the next RNA is the ribosomal RNA. The next RNA is the ribosomal RNA, and this ribosomal RNA together with proteins, they form the structural components of the ribosomes. And you have an idea about the structure of ribosomes. It consists of 2 subunits, a larger subunit and a smaller subunit. And these ribosomes are the sites of protein synthesis. So during the process of protein synthesis, this mRNA, which has the genetic blueprint, or which is the genetic blueprint, will bring the message from the nucleus and take it to the ribosomes, which is present in the cytoplasm for synthesis or translation into proteins. Okay, so this ribosomal RNA together with proteins forms the structural components of the ribosomes. And these ribosomes are of different types in prokaryotes and eukaryotes. In prokaryotes, the ribosome is of 70 S types. The ribosomes in prokaryotes is of 70 S type, and in eukaryotes it is of 80 S type. The letter S refers to the sedimentation coefficient. So in prokaryotes, the ribosome is of 70 S type, and in eukaryotes it is of 80 S type. And as we have discussed, they are made of 2 subunits, larger subunit and smaller subunit. So the largest subunit in prokaryotes is the 50S, and it consists of 23S, 5S RNAs, and the smaller subunit is 30S, and it consists of 16S RNAs. So these are the three types of RNAs, 23S, 5S, and 16S RNAs are the three types of RNAs which are present in the ribosomes of the prokaryotes. While in case of eukaryotes, the largest subunit is of 60S, and the smaller subunit is 40S, and the largest subunit consists of 28S, 5.8S, and 5S RNAs, and the smaller subunit consists of 18S ribosomal RNA. So eukaryotes consists of four types of RNAs, 28S, 5.8S, 5S, and 18S RNAs. So similarly, like tRNA and messenger RNA, this ribosomal RNA, they are also transcribed from the DNA, and that sequence of DNA which transcribes this ribosomal RNA, it is known as R ribosomal DNA, and the chromosome region which contains this rDNA is known as the nuclear region. And this ribosomal RNA constitutes about 80% of the total RNA content of the cell, while messenger RNA, it constitutes only about 5% of the content of the RNA content of the cell. So this rDNA is the sequence which codes for this ribosomal DNA. So together with proteins, they constitute the structural components of the ribosomes, and they are involved during the process of translation. So besides this messenger RNA, or besides tRNA and ribosomal RNA, there are also other types of RNA like genetic RNA, where DNA is absent and RNA serves as the genetic material. As I've said, in case of many of the plant viruses and animal viruses, RNA is the genetic material. And then there is also small nuclear RNAs, where besides the genetic material or besides DNA, RNAs are also present, which helps in the replication of DNAs, and those RNAs, they are known as small nuclear RNA. And the other is ribozymes. So these ribozymes are special RNAs which act as biological catalysts, and as name implies, they help in the formation of peptide bond during the process of protein synthesis. Okay, so genetic RNA, small nuclear RNA, and ribozymes, there are some types of RNA besides these three types of RNA. So in the end, let's quickly see these types of RNA. So RNA is a single-stranded polynucleotide chain which consists of ribose sugar, pentose phosphate, and the four types of bases: adenine, guanine, uracil, and cytosine. And it is of three types: messenger RNA, which is the blueprint, which contains the blueprint of the genetic information, and it constitutes about 5% of the total RNA content of the cell, and it carries the information from nucleus to the cytoplasm. And tRNA, which is also known as transfer RNA, it's responsible for bringing the amino acid from the cytoplasm, and by reading the codons on the RNA at this anticodon region, and it constitutes about 10 to 15% of the total RNA content of the cell, and it, due to the folding, intramolecular folding, it exhibits a clover leaf structure. And ribosomal RNA is has the highest percentage of RNA in the cell. It's about 80% of the total RNA content of the cell, and together with proteins, they form the structural components of the ribosomes, and it's of 70S, it's of different types in prokaryotes and eukaryotes. In prokaryotes, it is of three types, and in prokaryotes, it is of three types, and eukaryotes, there are four types of ribosomal RNA. And besides this, messenger RNA, tRNA, and ribosomal RNA, they are small nuclear RNAs. Then there's the genetic RNA and ribozymes, which takes part in different function of the cell. So all these types of RNA, they do not function in isolation. They do not function in isolation, but they function in a coordinate manner in carrying this genetic information from the nucleus into the cytoplasm. That is done by the messenger RNA, while during the process of protein synthesis, the amino acid is brought from the cytoplasm to the sites of protein synthesis by this transfer RNA, and this ribosomal RNA, they are responsible, together with the proteins, they are responsible for forming the structural components of the cell. So in the next class, we will see what does these three functions, how do they perform this function of transcription and translation in detail. So I hope to see you, and please come back again. Thank you.